The Lowiaceae and the Strelitziaceae are two families in the charismatic order Zingiberales. The order can be separated into two main groups of 4 families each. The 4 families around the ginger family, the Zingiberaceae, and the 4 families around the banana family, the Moussaceae. When we look at phylogenies of the order that have been conducted, we see that the phylogeny of the banana groups is very labile. Almost every phylogeny that is run shows a different relationship among the families of the banana groups, whereas the phylogeny of the ginger groups of families is very stable. It's always the same, with the Zingiberaceae sister group to the Caustaceae and the Canaceae as the sister group to the Marantaceae. One of the few things that is stable among the phylogenies of banana groups is the relationship between the Lowiaceae and the Strelitziaceae. Most of the phylogeny show that these two families are sister groups. Today we're going to look at a couple characters that might support this phylogenetic relationship, and maybe even start to solve some of the other problems in the phylogeny of the banana families. The Strelitziaceae is a charismatic group of species, including such things as Phenacospermum, shown here, Ravenella, the well-known traveler's palm, and Strelitzia, of which there are 2 main groups, one consisting of large, almost woody herbs with lateral inflorescences, and the other rhizomatous herbs, a little bit smaller but certainly not small, with inflorescences that arise from the axes of the leaves and consist of a single bract, whereas things like Strelitzia nicolai have multiple bracts in their inflorescence. The structure of the inflorescence in these 2 groups of species is, however, very similar. The Lowiaceae, on the other hand, is a small group of tropical herbs scattered throughout Southeast Asia. None of them are over about 1.5 meters tall. If we look at the base of the plant, we'll find the flowers, this one going into fruit with its persistent calyx. Careful removal of this floral complex shows that it has a structure like this. Here are 2 flowers borne on 2 very closely related axes and subtended by a continuation shoot. The continuation shoot will continue the growth of the rhizome and will eventually produce another group of flowers like that one shown here. Careful dissection and study of development shows that the structure of this inflorescence is like this. It has a single terminal axis which aborts. There are 2 scale leaves, and in the axil of each of those scale leaves, an axis arises. There are several bracts borne on that axis, and then there is a floral bract subtending a single flower. If we look for this aborted apex in the shoot itself, we would find it down between the 2 flowers, and it is sometimes visible there upon careful dissection. In addition to that aborted main apex, there is another apex that aborts, and that is, at the top of each of these shoots, there's an apical meristem that stops growing after the production of this first bract and flower. We can see that if we look at development in Orchidantha chinensis. Here is the flower, and there is the aborted apex. If we looked at Orchidantha maxillarioides, we would see that this apex grows slightly beyond this flower and produces several rudimentary bracts. These bracts, however, never open and never produce a mature flower. But still, we can draw out these bracts, and perhaps one other one, to show the idealized structure of this inflorescence. In this case, we would have a number of primary bracts, or main bracts, each subtending a single flower. Well, there's one other place where we have abortions in this family, and that is in these single flowers. Within the Zingiberales, whenever we see a single flower, it is always a reduction from a cincinus, or a monochasium. So we can put a monochasium back in the position of these single flowers to get something like this, as our idealized inflorescence structure in the Lowiaceae. Once we've done this, we suddenly start to see a great similarity between the inflorescence structure in the Lowiaceae and that of the Strelitziaceae. Here's a diagram of inflorescence structure in the Strelitziaceae. Instead of scale leaves, we have full, mature foliage leaves. And instead of an aborted apex, the apex continues to grow and produces new inflorescences, each in the axil of a leaf. The older inflorescences senesce and fall. There are usually 1 or 2 inflorescences open at any time in a mature flowering shoot, and there are several inflorescences that are developing in the higher level leaves. When we look at Strelitzia and Ravenella, we can see this inflorescence structure with the central axis which continues to grow, and then the lateral inflorescences produced in the axils of the foliage leaves. So there's a good deal of similarity between the inflorescence of Orchidantha and that of the Strelitziaceae. They're built along the same kinds of patterns. Wherever we're going to consider this now in an evolutionary context, we need to think about what the structure of the inflorescence is in the other families of the Zingiberales. And when we look at that, at least in overview, we see this. All of the other families have a simple inflorescence which consists of a few sterile bracts and then a number of floral bracts, each of which subtends a syncytium. Now I'm oversimplifying this a little bit, especially in the case of the Zingiberaceae, but I don't believe I'm distorting the structure. This is the basic structure of the inflorescence in all of the families of the Zingiberales, except for the Lowiaceae, the Strelitziaceae, and the Marentaceae. And the Marentaceae is a very special case that is very highly derived, but it is based on this same kind of pattern. So that suggests that we have a good character here. We have a character that unites the Lowiaceae and the Strelitziaceae. They share a common inflorescence architecture. This all looks very good until we remember that Phenacospermum has a single main terminal inflorescence, very much like we find in the rest of the order. We can see that very easily in the mature plant of Phenacospermum. Here is the main inflorescence axis. It's terminal on the shoot, and it bears lateral bracts, each subtending a syncytium. So the structure in the Strelitziaceae is really variable. We find both complex inflorescences, which are very similar to what we find in the Lowiaceae, and we find simple terminal inflorescences like we would find in the rest of the order. Can we still consider this inflorescence structure as a uniquely derived character in the Lowiaceae and the Strelitziaceae? And I think if you think about it a little bit, you would see that it is quite reasonable to draw that conclusion. This conclusion is supported when we look at Krohn et al.'s recent phylogeny of the Strelitziaceae. We see in this group that Phenacospermum comes out here as sister to Ravenella, and thus, if we were to plot any characters on this tree, the characters of the inflorescence structure, for instance, we would see that the structure of the Phenacospermum inflorescence is a reversion, or even more correctly here, is a uniquely derived character for Phenacospermum within this group. So we have, it looks like, a good character uniting Orchidantha and the Strelitziaceae. But what about this problem here with Strelitzia reginae and its relatives, which do not have the same kind of branched inflorescence that Strelitzia nicolai has? Here they are again, Strelitzia nicolai, having that inflorescence structure which we've just talked about with our lateral branches, our lateral inflorescences. But as I've said, this single bract of Strelitzia reginae and of the other species which are related to it is identical to these bracts which are on the inflorescence of Strelitzia nicolai. They both bear syncytium. It's just in this case that we find only one bract, whereas in this case the axis bears multiple bracts. They are both axillary, in this case on an upright shoot, and in this case on a rhizomatous shoot. In fact, there are cases where you can find in Strelitzia reginae an axis which bears not one, but two bracts. And again, this suggests that we're dealing with exactly the same kind of inflorescence structure in Strelitzia reginae as in Strelitzia nicolai. So these two inflorescences, which look so different when we first see them, the inflorescence of Orchidantha and the inflorescence of Strelitzia, are constructed on the same basic plan. and they argue for the monophyly of the Lowiaceae and the Strelitziaceae. That is, they support many of these phylogenetic analyses which have placed these two families together. I want to look at one other characteristic before ending, and this characteristic has to do with flower development. We're going to look at the development of the third sepal, first in Orchidantha and then in the other families. So here we have Orchidantha chinensis, and we have the development of the three sepals, number 1, number 2, and here number 3. It's delayed in its formation. Later in development, we can see that the third sepal remains smaller than the others, still later, still small, and right up to the point where the sepals are closing over the flower, the third sepal remains smaller. So the third sepal is delayed within development. We find the same thing in the Moussaceae. Here we have the first two sepals formed, and the third sepal has not formed at this time, and in fact will remain smaller for the early stages of development. The same thing is found in the Strelitziaceae. Here's Phenacospermum with the first, second, and third sepal delayed. And the same in Strelitzia nicolai. This is the first sepal, the second, and the third. Again, the third sepal is delayed in its formation. How about the Heliconiaceae? Well, the Heliconiaceae has a different state. Here we see the syncytium of Heliconia with the flowers. This is the first formed sepal. This is the second. This is the third. If we look at earlier development, first, second, third, there's no delay in the formation of the third sepal. So there seems to be a character that is uniting the Strelitziaceae, the Lowiaceae, and the Moussaceae that is not found in Heliconia. What about the other families? Well, initially it looks pretty good because in the Zingiberaceae we find that there's no delay between the formation of the second and third sepal. Here we have the first, second, and third, no delay, first, second, and third. This flower is a mirror image of the previous one. Again, no delay though. And in globa down here, again, first, second, and third, no delay in the formation of the three sepals. Unfortunately, there are other species within the ginger groups, and they do have a delay in the formation of the third sepal. Here we see in the Caustaceae a very strong delay in Teponoculus. In Costus scabra, the same thing, third one delayed. Alpinia, again, third sepal delayed. And in Zingibera zerumbit, the third sepal is delayed in formation. In the Marantaceae, well, no one has really looked closely at this. My own work on the Marantaceae was done long ago, and I don't have good enough photographs to suggest definitively whether the third sepal is delayed or not. These diagrams suggest it is not strongly delayed, but I think it needs investigation. However, I would argue that knowing the state of this character in the ginger groups is not essential. Certainly, we're beyond the stage where we expect all families to, in an order, to have the same kinds of character state, that is, to be little variability. We've seen many cases of reversal and parallelism. And I think what we've got here in the Zingiberaceae and the Marantaceae is a case of parallelism or reversal. But the fact that we have species within this group which do not show a delay in the formation of the third sepal, and that we have that same state occurring down here in the banana families suggests that that could be a primitive characteristic which would unite then the Lowiaceae with the Strelitziaceae and the Moussaceae by the character a delay in the formation of the third sepal. And this is one of the very few morphological characters we have that may be useful in working out the phylogeny of the banana group. And I'd just like to thank my co-authors on this paper from the South China Botanical Garden, Wangfang Liu and Jingping Liao, and then some of the gardens where I've collected the material, Henning Kunze for taking that picture of Tapinochylis, and Max Dulin for taking many of the photographs of Musa development. Thank you.